Welcome everyone. My name is Jihan Abdurrahman, and today we will explore an important topic about cell cycle regulation to make sure the proper cell division and growth. We have 4 points in our agenda today. Number one, introduction, cell cycle phases, regulation of cell cycle, and last point, how cancer happen during cell division. In introduction, we need to know what is cell cycle as a definition. Cell cycle is a series of repeated events which happen inside all eukaryotic cells. There are three reasons make this cell cycle important. First is growth. The cell needs to make this cell cycle for growth to increase size. All multicellular organisms want to make sure that there is increasing in the number of cells during the cell division. The second reason for cell to make it very important to repair any damaged cell or any dead cell to replace through this mechanism of action inside the cell. Number three, for reproduction. So all the cells need to continuity of life, to reproduce and dividing, and this is happened through the cell division. The second point in our agenda, what are the phases of cell cycle? There are many important phases in the cell cycle. Actually, we have 4 important phases. The first one is interphase, which is the longest stage in cell cycle. Interphase include G1, S, and G2. So each stages of these consider as a specific part which has some event happen inside this stage. The third stage or fourth stage is mitosis. Mitosis, or the word mito mean division of nucleus. So in this stage, or by the end of this stage, we can find 2 nuclei inside the cell through 4 important processes: prophase, metaphase, anaphase, and telophase. So over here, we can find the cell move through a series of phases in the order manner during interphase G1, S, G2. Let's see what's happened in each stage of these. So in G1 or gap 1, the cell grow in size and the protein start to form and all organelles start to form inside the cell. And this is preparing for the DNA replication. In S stage, this stage very special to the DNA or DNA synthesis. So we can find the DNA replicate or make copy of itself. So by the end of this stage, we can find 2 identical copy of gene. So the chromosome will duplicate it to make sure that the daughter cell receive the complete set of genetic material. In gap 2 or G2 phase, the cell continue to prepare for the division, and the protein and organelles that require for cell division start to form. And now the cell and interphase or introduction for cell division. The fourth stage, which is metaphase. In a metaphase, go through four important stages. The first step of mitosis is prophase. So in prophase, we can find the chromosome condense, the nuclear membrane dissolve, and this chromosome or genetic material start to condense more and more in prometaphase, and the spindle fiber start to form. In metaphase, we can find the chromosome line up at the center of equator, and the spindle fiber become longer comparing with prometaphase. And when the chromosome line up at the center of equator, cell prepare for this division by moving the chromosome to the opposite or chromatid to the opposite pole of the cell, and this stage is called anaphase. In a telophase, the nuclear membrane reform around the nucleus, and now we have two identical sets, a set of chromosomes in each margin of the cell, and this stage called telophase. After telophase, now the cell need to divide the cytoplasm, and the cytoplasm start to divide in a process called cytokinesis. And the cytokinesis, it's different from plant cell to animal cell, because in plant cell we have a cell wall, but in animal cell we don't have any cell wall. So in an animal cell form something called clavicule, but in plant cell we form cell plate due to presence of the cell wall. And this in plant cell controlled by Golgi apparatus and vesicle, which align at the center of the cell wall to form the division. Here is a brief or summary for each of these stages in mitosis, like prophase, what's happened, metaphase, and anaphase, telophase, and cytophase. Because the cell division is very important, we need some enzymes and protein to control this kind of cell division. Number 3 is regulation of cell cycle. This is the role of the enzymes and the protein which control the cell cycle. We have many events need to control each stage of the cell cycle by using the protein and the enzyme inside the cell. The enzymes and the proteins which control the cell division is called cyclin and cyclin-dependent kinases. The difference between cyclin and cyclin-dependent kinases, that cyclin is, or cyclins are protein that play an important role to control transition between different phases of cell cycle like G1, S, G2, and the cyclins are synthesized at specific point in a cell cycle to ensure the movement of the cell is controlled manner. The second kind of protein, which is cyclin-dependent kinases, it's called cyclin-dependent because it's dormant most of time, but it start to activate depending on the cyclin. So this is why we call this cyclin-dependent kinases. So cyclin-dependent kinases are the enzyme or protein which is inactive and require binding to the cyclin to become more active. Protein kinases such as cyclin dependent kinases are has very important role and are vital in the sequence of activities of cell cycle. Why? Because they are responsible for phosphorylation key protein for transition of phases like G1S and G2 and mitosis. Okay. In this picture, we can find how cyclin protein and cyclin dependent kinases are working together and bind and start to activate. So cyclin over here is activated cyclin dependent kinases by phosphorylation and start to activate the target protein to regulate the cell cycle. After that, the cyclin destroyed. In another picture, in this picture, we can find the relationship between cyclin level and cyclin dependent kinases activity. So we can find the peak of cyclin dependent kinases in a mitosis is the highest, and in interphase start to accelerate over here and get down at the first beginning of interphase and then raise up during the end of interphase and mitosis. Because cyclin and cyclin dependent protein is very important to the cell cycle, so we must have a checkpoint during each stage of the stages of cell cycle and before we start any of these stages. So checkpoints are very critical and control point that ensure the cell cycle proceed correctly. In first stage, we can find this checkpoint in many of these cycle, like in a four times. We can find this in a 4 times. So we can find the first checkpoint in G1 and S checkpoint, which determine whether the cell should enter S phase and check the cell size, nutrients, growth factor is enough, and if the DNA damage or not. So we can find this checkpoint between G1S. So over here, we can find the first checkpoint. Like, for example, when you're going from New York to New Jersey, you have some checkpoint, like traffic light, to make sure that you have enough gas or not. So this is which is happening exactly inside the cell cycle. So the first stage or the first checkpoint in cell growth, which happened toward the end of the gap to check if the cell is big enough, if the cell has made the proper protein for synthesis phase, or if the cell go through the resting period or not. But the question now, if the cell doesn't have any of these, doesn't have enough protein or growth factor, so what's happened during this phase? At this stage or at this process happen. So we can find something called G0 phase. G0 phase is the resting phase where the cell exists or stop dividing. Some cell like neuron or muscle cell enter this phase directly, undergoes the division again. This phase is very crucial for conserving energy and the resources in non-dividing cell. So most of the cell and muscle cell stay in the G0 phase to make sure that they are still in a resting phase. Okay. The second point after G1 phase, we can find G2 phase or G2 checkpoint. In a G2 checkpoint, this is before the cell enter to the mitosis. At this stage, we make sure that the DNA replicated and this replication process complete to prevent the cell with any damage or to enter the mitosis with the complete copies of DNA. In the M stage, the spindle, we call it a spindle assembly checkpoint. We want to make sure that the spindle fiber or the spindle thread is enough to make the mitosis division. Because the one which moves the chromosome is the spindle fiber. So we need to make sure that the simple fiber completely assembly and the chromosomes are proper attached to this spindle fiber before segregation or separation. So in a brief here, these are the main checkpoint. G1 checkpoint to ensure enough energy, growth factor, and nutrients. If we don't have it, the cell will exit to the G0 phase. And G2 checkpoint, before intermitosis, we need to make sure that the DNA synthesis and the DNA copied completely and properly in S stage. The last stage is, the last checkpoint is M checkpoint. In M checkpoint, we need to make sure that spindle assembly checkpoint complete and it's attached to the chromosome. What the relationship now? The question: what is the relationship between cyclic dependent kinases and kinases or cyclic enzymes and this checkpoint? So we can find 5 important phases over here. Each one has a strong relationship with cyclin and cyclin dependent protein complex. So in G1 over here, we can find the cyclin D, CD, cyclin dependent kinases 4. The main function of cyclin D and cyclic dependent kinases 4 to prepare the cell for DNA replication. Why? Because the next stage is S stage. So this is which is happening in G1. Between G1 and S over here, G1 and S, we can find the second enzyme, the role of second enzyme, which is cyclin E and cyclin dependent kinases 2. This enzyme or this protein is commit the cell to S stage fully inactive. After that, we can find the DNA synthesis, and the DNA synthesis in S stage. So it's initiate and complete the DNA replication. In G2, the cyclin A and C cyclin dependent kinases 1 prepare the cell for mitosis, and between G2 and M, we find cyclin B or cyclin dependent kinases 1, which enter into mitosis and regulate chromosome segregation. The question is, what happened? What's happened if the checkpoint failures? What's happened if there is any dis or damage in the cell, and we don't have enough protein, we don't have enough DNA, there is no growth factor? What's happened? At this time, if the cells are damaged beyond repair or proper cell cycle regulation, this can trigger something called cell apoptosis. Cell apoptosis, or this is called program for cell death. This is very crucial mechanism to eliminate the potentially cancer cell and maintain overall tissue health. Or there is something happen if the cell does not go through it, so we can find cancer uh, happen to the cell. What is cancer? Cancer is irregular of cell division. So the cell start to divide and divide and divide rapidly in abnormal way and become less dependent on the signal transduction in from the cell from other cells. So we can find a different kind of cancer have different mutation signature. However, the scientific comparison of multiple tumor type has revealed that certain genes are mutant in cancer cell more often than other. But this cancer cell we can discover it in an early stage. At this time, we call it tumor as long as it's still inside the place or there is no leaking from any of this abnormal cell to the bloodstream. Once the one of these cells leak or escaping with the bloodstream and affect or invade another cell, at this time we call it cancer. So the cell sometimes protect itself by using this death program. This death program is very important to protect the cell and eliminate any cancer cell and to maintain overall the tissue health, body tissue health. We have many resources here. This resource very important. Some of it from the book, like the last 3 books, and the other with a citation. So thank you so much for watching my presentation. And if you have any question, just write this in the classroom discussion and we can discuss it together. Thank you so much. And I appreciate your time and your watching. Thank you.